Hello everyone, and welcome to today's live broadcast, Performance Assessment of RNA Sequencing and Expression Arrays for Transcriptome Analysis in Cancer Research, presented by Petr Nazarov, Research Scientist, Bioinformatics and Modeling Group, Proteome and Genome Research Unit, Department of Oncology, Luxembourg Institute of Health. I'm Alexis Corrales of LabRoots, and I'll be your moderator for today's event. This educational web seminar is brought to you by LabRoots and sponsored by Thermo Fisher Scientific. For more information on our sponsor, please visit thermofisher.com. So, let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, click on the arrows at the top right-hand corner of the slide window. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Answer a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I'd like to now introduce our speaker, Dr. Petr Nazarov. Dr. Petr Nazarov is a research scientist in the Proteome and Genome Research Unit of the Department of Oncology at Luxembourg Institute of Health and an invited assistant professor at Luxembourg University. Dr. Nazarov produced over 30 publications devoted to data analysis and oncology. His primary expertise is biostatistics, machine learning, and genomics. His recent work focuses on characterizing transcriptional variations in peritumoral tissue environment of pancreatic cancer, as well as tumors originated from lung, colon, skin, and brain. His current research leverages deconvolution of transcriptomic signals from heterogeneous tumors to improve cancer classification. Dr. Nazarov, you may now begin your presentation.
Thank you, Alexis, for this nice introduction. Hello, everyone. So today we will speak a little bit about the performance assessment of RNA sequencing and expression arrays for transcriptome analysis in cancer research, but not only about this. In fact, I have two parts in this presentation. We will start comparing RNA-seq and microarray performance. So we shall see how similar the results are, what are the specific features of both platforms. When you study coding and non-coding genes, for example, and what kind of strange things we observed looking at the alternative splicing by these both techniques. And for the second part, I will briefly introduce you a method which is used in transcriptomics, which can be used in transcriptomics for deconvolution of complex mixed signals from different cell types, and try to convince you that it's a great method which can help us understanding more about cancer. So let's begin then. And the first part of the talk is based on the paper which we published just last year in BMC Genomics. So if you have more questions about it, you can went through this and have a look. Next, let's go. So why we're interested in comparing different platforms? In literature, usually people claim that RNA-seq is outperforming array techniques, and well, for some situations that's true, without any question, but we were a bit puzzled because in majority of cases, people tend to take quite old microarrays, like, for example, Affymetrix U133 arrays, which are 3' IVT, which have only prop sets in its, let's say, 3' of each transcript, and compare it to quite new technique, which is RNA sequencing. So we would like to see what would happen if we take the modern arrays like HTA version 2, by the way, now there is a successor of it called Clariome D arrays. So what will happen if we take these modern arrays, which have probes through all the transcriptome, including the junctions between different exons, and compare it to RNA-seq. In our view, such kind of comparison would be more fair. So we are interested in how the results are similar in terms of expression, what is the correlation between protein coding genes, between other biotypes, between exons, what differences can we observe with these platforms, and ultimately to find some suggestions, which platform should one use if he's doing research, if he's doing some kind of standard analysis, and so on. What we've done, we took 9 patients with lung squamous cell carcinoma. The data were presented to us by the French institution, Unité en Serme, University of France, and we took from these patients, from each one, 2 samples. One was taken from the tumor core, another one from adjacent normal tissue. So in total, we had like 18 samples. Those samples were processed in a standardized way and analyzed by Affymetrix HTA version 2 arrays. We used 100 nanogram of the RNA material there, or with Illumina HiSeq 2000, where we used from 500 to 500 nanograms to 1 microgram. I should mention that in this situation, for sequencing, we use a poly selection. This might be important to see the results, to understand the results, and we can only target with this technique then those genes which have poly-A tail. This is something for us to remember. In the sequencing, we aimed at library size of 2 million base pairs. So what we got after alignment, after mapping, we got like from 120 to 280 million base pairs. Million reads, each one composed of 77 base pairs per read. In general, we saw that normal tissue, which is represented here by green color, is a little bit more homogeneous in number of map reads. For cancer, we had more variability, the orange color here. Just to describe in brief what kind of method we used, what kind of strategy we used, I prepared the next slide. So for arrays, we went through a kind of standardized pipeline. We took the cell files, we analyzed it with Partiq genomic suite, which can be, for example, replaced by transcriptome analysis console if you don't have the access to the software. So the normalization was performed and probe summarization within the tool. We use RMA for normalization with GC contact correction. Afterwards, we continue with probe set expression, and these probe sets were in fact remapped on the genome in order to be able to characterize as many genes found in RNA-seq as possible. For RNA-seq, we started with FastAQ files. We perform alignment with top hat, and then the BAM files were analyzed either with HT-seq for gene expression or with feature count from our bioconductor. It's the method, the function, let's say, from the R subreads, in order to count exons. Unfortunately, HT-seq was not able to count exons. There are some technical issues with this. So next, let's see what we get in terms of characterization of the genes. We took two groups of genes for consideration: the protein coding genes, those which are standard, well known, and also the non-coding RNAs, long non-coding RNA, where we combine together several subtypes, which you can see on the left. So those subtypes which are here represented by the green boxes are combined all together and form the group of long non-coding RNAs. Just to remember, we have poly-A filtration. That's why we have only those RNCs which still have the poly-A tail. The coverage after remapping of HTA was amazingly good. So we got like over almost all protein coding genes were found back. The vast majority of LNCs were found back as well. And this works both on the level of genes and on the level of exons. Below you have the Venn diagrams for the level of exon overlap between the two platforms. Next, if you would like to compare the genes, compare the data, one of the way would be to use something like dimensionality reduction method, principal component analysis, for example. And if you take the data as they are, of course, you will see all the differences between different scale of the data in RNA-seq and in microarrays. That's what's represented here. We see two big clusters. Fortunately, this kind of bias is consistent and can be removed, for example, by simply subtracting mean and dividing by standard deviation. So by centering scaling, the data afterwards look much better. They form two clusters, normal and tumors, and we saw quite many similarities in both NPCA and also by the heat map on the right between the same samples studied by both techniques. For non-coding RNA, for LNC RNAs, we saw approximately the same picture, just with increased noise, basically, but still the same two clusters were found. Next, we checked for the correlation between the genes, coding and non-coding. In terms of log signal, the correlation was 0.76. We used here Spearman correlation just to avoid the questions about the scales. So it's based on ranks. And for the fold change, it was like 0.74. So quite high correlation was observed between the signals and fold changes for coding mRNAs. However, for LNC RNAs, the correlation was much less exciting, let's say, 0.3 only. We started to investigate what was the reason for this and came out with the point that it seems to be linked to gene lengths. So on the left, you have here the scatter plot for one patient measured by both techniques, by HTA and RNA-seq, in terms of intensity of the signal, in terms of fold change between normal and tumor samples. What you can see is the most correlated genes are those colored here by red, right? And for those genes, the length was higher than 6,000 nucleotide. For those genes with length less than 2,000 nucleotide, represented by blue here, the correlation was quite ugly, I must say. And when we have a look at LNC RNAs, non-coding RNAs, what we can see is that the majority of these LNCs have lengths below 2,000 nucleotides, which means they are in the group of lowly correlated. There is quite low correlation. If you see carefully, you might see some red dots here as well, which as well show high correlation, but unfortunately the rest not. Next, as we see that there is quite low correlation for the genes with low expression, and we were not happy with the results comparing microarray and RNA-seq for non-coding genes, we start to find out what causing this problem. So length can be one explanation. We checked also the part of variability which can be explained by those factors which we can control in experiment, like patient-to-patient variability or tumor normal variability, so the effect of patient to effect of state. And we use the method called principal variance component analysis, PVCA, which showed us the part of total variability which can be explained by those two factors, and those part of variability which cannot be explained by anything. So those are residuals. We see here that the majority of variability come from tumor normal difference, and that's good. And also that the RNA-seq, which is in red here, show higher unexplained portion of variability compared to HTA. For link RNA, for LNC RNAs, their behavior was very similar, except that we can see much more unexplained variability in RNA-seq. So it leads us to idea that perhaps there is something not so nice in RNA-seq. We start investigating more about this. We checked what is the behavior of gene variability with its expression on this slide, and we saw that in normal tissue, the same as in tumor tissue, in both cases, RNA-seq show higher variability, especially for lowly expressed genes. This is represented by the red line, which is always above, so high variability, above the blue line is HTA. Based on this, we can tell that the majority of variability in RNA-seq come from those genes with low signal, small number of reads. Either it can be linked to length or to the expression level. Next, we go for the standard analysis, which is differential expression analysis. We use the HGR method for RNA-seq and LIMMA for HTA. What we found is that in most of the cases, we try different threshold, right? And with majority of the threshold, except if you take extremely low FDR, but for reasonable FDR levels, HTA outperform RNA-seq in terms of number of significant genes. Of course, the number of significant genes doesn't mean that the method is better. So we needed to compare with something, and we compare the data with TCGA, LUSC data set from TCGA. Quite many samples are included there. We found that the confirmation level is approximately the same. So HTA produce higher number of genes with a confirmation level about Jacquard index, for example, was 0.2. And RNA-seq produced less, but with the same Jacquard index. So number of confirmed features was also decreased, but the proportions stayed the same. Similar results were obtained with non-coding RNA. However, we were not able to confirm almost any of our non-coding RNA with TCGA because it's not oriented on non-coding genes, but some of them were found back. Next question which we asked was linked to what is the detection limits of each technique. We all know that RNA-seq have higher dynamic range. It's obvious, people speak about this, but what we propose in the paper is not to compare the data as they are, but rather to concentrate on the genes which are differentially expressed, which we just identified on the previous slide. So why not to look at those genes which are differentially expressed, consistent between different biological replicates, and see what can we tell about dynamic range. And that's the result here on this slide. The dynamic range is indeed higher for RNA-seq without an equation. It's 10 for RNA-seq in terms of log2 and 5 in terms of and 5 for HTA. However, if you look at the lower limit of absolute fold change which can be detected by the method, you will see that for HTA it's much lower. So RNA-seq produces you much higher variability in your data, but can detect reasonably high level of fold change, whereas HTA produce quite small variability, but also detect tiny differences in log fold change. So that's in fact an interesting finding, I think, telling that despite the dynamic range is high for RNA-seq, but there are some kind of also cost to pay for this high dynamic range. So we checked as well whether we can find the genes which can be used as predictive markers between cancer and normal. Of course, it's not very wise to try to find predictive markers in the group of only 9 patients. You need a much bigger cohort, but we tried just to see the tendency, and what we saw is that in coding and non-coding genes, you see more genes with area under the curve above 0.95 in HTA. For example, so here you have distribution of all the genes characterized by their area under the curve. AUC, which goes from 0.5 for completely random situation to 1 for ideal classifier. So you can see that HTA in this kind of test outperformed RNA-seq with number of the genes with predictive power, both coding and non-coding. We tried to confirm the results with TCGA, and we found basically a very similar number of number of confirmed genes. So confirmation level was quite stable. Next, we checked what kind of interesting things we can find if we feed now the significant genes to enrichment analysis tool. If we try to find some biological process which are enriched, I have some issue with my computer. That's strange. I think it's solved now, I hope. So whether we can find the same biological functions looking at the significant genes in RNA-seq and in HTA. In fact, the strongest signals and the most important biological processes which we investigated and which are enriched with the significant genes, they are the same. It's the cell cycle, for example, ciliary organization and the movement, which is very specific to lung tissue, cell division, and so on. However, there were some biological processes which are detected only in a specific platform. For example, in RNA-seq, we saw more biological processes linked to extracellular matrix organization, for example, so something related to outside of the cell. And for HTA, we saw more specific things linked to DNA, so working inside nucleus. This was interesting finding for us. So we go deeper and check what happened with the cellular components, which are more specific. And indeed, with cellular components, we find the extracellular matrix with RNA-seq and internal part and something linked to DNA and nucleus, nucleoplasm with HTA specifically. Despite the fact that majority of cellular components were common between two platforms. So we tried to find out what can give us this bias. And what we've done, we took the genes which are which are involved in cellular components specifically identified with RNA-seq and specifically identified with HTA. So those red and blue line. Here arrows show that we take the genes from these components and check the expression level of those genes in RNA-seq experiment and in HTA. And RNA-seq is on the left, HTA is on the right. So exactly the same genes were checked in two platforms, whether we can see something similar. In fact, we saw a similar feature. We saw that the genes which are unique for RNA-seq based components, they are always higher expressed than those which are unique for HTA. So HTA a little bit lower expressed. We checked also whether we can find the same with biological processes, but no, it was specific to cellular components, despite the fact that biological processes as well show some bias, but we were not able to find those specific genes there. So cellular components play a role. And we also check whether we can explain this bias by the length variability of the genes, which are working in nucleoplasm and extracellular matrix. No, we were not able to confirm. Next, we try to correct for the bias which can be linked to the length effect. The shorter genes are harder to characterize with RNA-seq because just they are shorter. So we use a specific tool called GeoSeq to correct for the effect of the lengths. We got no improvement, or almost no improvement. Let's say we were able to detect the same bias. So it cannot be explained by the lengths, but rather by the abundance of the RNA. It seems that those genes involved in production of protein for extracellular matrix are more abundant than those which are working afterwards in the nucleus. And that's it with the gene expression analysis. Next, we go for analysis of splicing events. And here we use the two levels of information. First of all, we use the expression of the exons, and second, we use the expression of the junction between the exons. Both techniques can give us this information. We started first trying to use HT-seq for exons. It doesn't work just because there are many overlapping exons in genome, so they are not going to work properly. Don't try, even, I would say. HT-seq is for genes. It works greatly for genes, but not for exons. For exons, we used feature count from R-subread package. And the correlation was, again, quite high. If we start comparing the level of the signal between two platforms, the correlation of the signal was 0.6, 0.7, a little bit less than genes, but still reasonable. However, as soon as we went for the analysis of differentially used exons or splicing events, we found a very strange picture. We saw that the overlap is almost random between two platforms. Well, it's not exactly random, it's more than random. We checked the statistics, but it's still very, very low. So like 3.4 thousand common and between 23 significant exons found in RNA-seq and 27,000 significant exons found in HTA. We checked also the junction. For junction, we got much lower number of significant in the RNA-seq. Still, overlap was very poor. Afterwards, we tried to combine for each platform exon and the junction together in order to have something confirmed from both levels. We got 3.3 thousand RNA-seq found, exons found by RNA-seq, and like 12,000 found by HTA, but still the overlap was very low. What can be the reason of this? We looked on this and find out that it seems to be linked with two things. First of all is the location in the transcriptome. If you look on these curves here, those represent probability density of a splicing event to be detected in HTA and RNA-seq, depending on the location in the transcript. So from 5' to 3', taking into account all the data. So you see clearly we have a peak with RNA-seq at 3'. Those exons more often found to be significant in RNA-seq. And also we found that the longer exons, the red line here, longer exons in RNA-seq have tendency to be more often found like differentially spliced. So there are like two reasons. We checked the data, quite many genes one by one. The general tendency was like this. This is just two example, two genes. On top you have HTA and RNA-seq for one gene in the direct strand, and below in the reverse strand. So what you can see is that with RNA-seq we always have increase of the signal intensity to 3' end in both cases, and quite stable results for HTA. It of course could be linked to the fact that the last exon in genes tend to be longer, which you can find out with whatever database. However, even if we corrected for this bias, we were still able to see some discrepancies, so it doesn't correct the results at all. So we still saw quite low overlap. And on the lower picture, for example, you still see that in the terminating exon at 3' you have much higher, or a little bit higher still, number of significant exons than in other parts. So with this, I would like to conclude the first part of the talk. In our study, it seems that HTA really show more reliable results than RNA-seq with 200 million reads. Just to remind you that the study was performed around, I think, over 5 years ago now, or around 5 years ago. So maybe now the situation has changed, but so far what we see, we can confirm this result with other studies. We still see the same problem there with the variability. Second point is that gene length sensitivity makes RNA-seq a difficult technique for non-coding RNA, meaning that as soon as you would like to characterize short RNAs, short transcripts, you should have much higher coverage, or you have to specifically select those short transcripts by something. Otherwise, all your reads will be coming from the longer, more expressed genes. Still, RNA-seq is a very nice discovery tool, so it's impossible to study new transcript, new regions which are transcribed unpredictably without this tool. Formation of chimeric RNA, chimeric genes, it's also impossible. So this should be, of course, noted. And the point which I would like to bring to you, be careful when you do isoform study with whatever platform. At least with our experience, there are some issues, and the overlap between different tools is quite low. So be careful and try to validate the results with other tools, with other methods. So that's it for the first part. And now we should go for the second part. Please give me 1 second. In the second part, we are going to speak about the method called independent component analysis in the transcriptomics. This is a project which I'm interested in a lot and in which I'm working for the moment. And to start with, let's begin with this strange slide. This is a visualization of the cocktail party problem. Imagine all these people standing together with music playing, having drinks and speaking. And we are particularly interested in what James is telling. So how can we solve it? It's not an easy task, as you can see, because you will get the sound from all different sources. And in fact, this is exactly the situation what we see when we try to characterize tumor tissue by taking a bulk sample, a biopsy. In this biopsy, you are going to have quite many cells from various origin. It can be in this situation, you can see you have endothelial cells, the normal cells in the stroma, the fibroblasts, immune cells, and the cancer cells in which we are interested. And what you're going to get is a mixture of all this transcriptome in your sample. Of course, you can make the sample, to purify the sample, to have like from 80 to 90% of cancer cells, but still the signal of immune cells, for example, usually is not neglectable. It's almost always there, and your transcriptome will be still mixed because you can have several subtypes of cancer cells. Fortunately for the first picture, the solution was there, is there for quite many decades, I think. And the method is called independent component analysis. What you should do in this situation, you should get as many linear combination of this mixed signal as you can, and you should run the method, which is very similar to PCA, just with different criteria, which will separate you the original signal into statistically independent components, statistically independent signals, and then you can recognize what one person was telling. So how is the method work? In just a few words, you should take the samples from quite many patients, as many as possible. And this is represented, of course, you can sequence it or do microarrays with it. So as a result, you get the quite large matrix of original data with many samples and like 20,000 genes. The method performs matrix decomposition, so it's transformed this into a matrix product of two matrices. The first one we can call, for example, metagenes. It is in space of genes and number of components. And the second one is how each component is represented in each sample. So the first one shows how each gene is involved in each component, and the second one is how components are involved in samples. The genes are statistically independent. You can see that there is no pattern at all in this metagene. And only few genes usually have an effect on each component. And those genes can be analyzed with standard methods like with enrichment analysis, and you can link them then to biological processes. You can link components, independent components, to specific biological processes. The weight of each component within the cohort of patient can be linked to some clinical observation, for example, subtype of cancer or survival of the patients. If you would like to know more about this gene, I recommend the two papers below, which you can have a look to get more information about the method itself and about its application. So, just a few hints about what the method can do and what it cannot do. The positive things about the method is that it can find statistically independent signals in the expression profiles. Based on these independent components, you will be able to find the most important gene within each component, and thus you can link it via enrichment analysis to biological processes. The method can tell you what is the weight of each component in the sample, and so linking by this you can link it to clinical data, to clinical observation. The method does not need, in fact, to be run any additional knowledge, so you don't need to know the profile of subtypes of cells, like subtypes, the profile of leukocytes, of stromal, or of other stromal cells, of cancer cells. In order to make the deconvolution, you can use the data as they are, just one requirement that you need a lot of data. The answer is quite robust. If you repeat your analysis, you will get the same results, or almost the same results, except that no one tell you whether the first component is going to be first in the next analysis. Maybe the first component will appear as the third component or the 10th component. But otherwise, if the component is found there, most often this is the quite robust component. There are, of course, some contrast, some drawbacks of the method. There is no ranking of the components. Not like in PCA, you cannot tell that the first component is more important than the second one. The results are still not deterministic. So there is some random noise, some randomness. Some components are always found, like in 99% cases. But there are some components which sometimes found, sometimes not found. They're not so strong. Maybe they can be neglected in the analysis or removed from the analysis. The orientation of signal is arbitrary. So if component number 1 in first analysis was linked positively to immune signal, then in the second analysis of the same data, you can find there is component number 3 now called, and it's a negative part of this component. So we should take into account this possibility of double orientation. And the method is not perhaps the best one if you look for precise estimation of cell fractions. You will find the correlation with cell fraction, I'm pretty sure, at least we found it quite often, but maybe it will not give you a quantitative results. At least we were not able to get exactly quantitative result. Qualitative, yes, perfectly. So we use this method, we have an idea of using this method for patient classification. How can we use it? We can take the large cohort of data, like for example, coming from TCGA, take new patients, put them together, run ICA on this big data set, then using some machine learning, it can be random forest, for example, which work, for the moment for us the best. With random forest, you can train it on the TCGA data and try to predict the classes for the new samples, for the new patients. In addition to just classification, you will also get the knowledge about the biology happening underneath. You will be able to link each component to biological signal. So what we've done, we developed a parallel ICA which run in a consensus way. So there are many runs, multiple runs, excluding each sample in the data set, which provide quite robust estimation of both matrices, the matrix of the metagenes and matrix of the weights. And just to show you some kind of results we found, I would like to present you a few slides if I have time. We started working with this project on skin cutaneous melanoma from TCGA. So we took like all the samples available there, add 5 samples from our own, and try to see what it can tell us. So in addition to telling what kind of classes we expect for the new samples, we also checked what how good is the accuracy of the method if you use leave one out cross-validation on the training set, and the accuracy was quite reasonable. The gender was always found correctly, so there's no question about the gender. The type of the tumor, is it metastatic or primary, was predicted like for 79% of cases, and the subgroup of the tumor, the cluster of the tumor, was predicted with 90% accuracy, which is quite reasonable, I think. The nice thing about ICA is that it not only capture the biological signals from different cell subpopulation, but also can correct for the bias in technique or different platforms. For example, our data were jumping out from the distribution of the training data, as you can see it here on the left PCA picture. However, as soon as you perform ICA, the data were quite nicely incorporated in the distribution. There were some components which capture the differences between two platforms, and they keep this difference within, making the data available for further analysis. Another nice feature about the method that you can perform independent component analysis on several levels of data, like at mRNA, at microRNA, at protein, and combine them together into one cluster of different components, which are targeting exactly the same process. For example, we were able to find immune component at several levels, at mRNA, at microRNA, and the protein level, and they all were linked to survival of the patients. So it means that combining the information, the weight matrices from all these components together, you can build a kind of score which will characterize the immune signal in the patient with just a single number, which was nice. We tried this and we got quite nice confirmation. We were able to find the immune signal, the angiogenic signal there, the signal of keratinocytes, the signal of melanocytes, and we're able to characterize the new samples with this score. We also were able to combine together the hazards coming from the Cox regression model in order to be able to predict what is the hazard for each particular patient. So just to summarize, wrap up this, the second part, we tested our implementation of consensus ICA. The ICA is able to decompose the bulk data set into meaningful signals that we saw, not only with this data set, but also with other data sets. And the new samples are properly mapped into the space of independent component, not like with a PCA, but here you can really find them back with meaningful component, for example, signal of keratinocytes, and then you see how your samples are behaving there. And method allow classifying and scoring new patients, which is interesting for clinical research studies. So to the final slide, I would like to thank all the people who work with me in the first project and the second project, including our team here at the Proteome and Genome Research Unit at Luxembourg Institute of Health, but also from the Neuro-Oncology unit of our institution, which is led by Professor Simon Niclou, and also our collaborators from university, especially Dr. Stephanie Kreis, with whom we performed the study on melanoma. And a special thanks to Dr. Andrei Zinoviev from Institut Curie in France, in Paris, who is a very strong promoter of the method in Europe, I think, of the independent component analysis method. So thank you very much for your attention. Now I think we have time for questions.
Thank you, Dr. Nazarov, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Answer Question box located on the far left of your screen. We'll answer as many questions as we have time for. So let's get started. Our first question is, if the length has an effect on RNA sequencing results, why didn't you use expression measures that are corrected for the length bias, like FPKM or TPM?
Yes, thank you very much. It's a very good question, in fact. So what we observed is, we tested this, in fact. We tested this, and what we observed that using FPKM does not reduce the stochasticity. You just remove the lengths from consideration, but you still have stochastic noise, so you do not have many differentially expressed genes with small lengths. The first point is this. The second point is that in this situation, we cannot use any more models with binomial noise, with negative binomial signal, and we have to use lemma, so we get even smaller number of significant genes. Well, and finally, we did not see that there was any improvement in terms of gene ontology term bias. That's it.
Our next question is, can the bias be linked to a specific enrichment tool used? Did you use background list of genes during the enrichment?
Yes, thanks. For the second part of the question, absolutely. We corrected, we took into account, in fact, two reference lists of genes, those which are in general presented on the platform and those which are actually expressed in this tissue. And got very similar results, in fact. For the first part of the question, we tried 3 different methods, namely TOP-GO and GeoSec from R, but also online tool EnrichR. And in addition to this, we confirmed the results with Reactome pathway analyzer, Reactome PA, and we saw the similar bias there as well.
Our next question is, when are you planning to publish the results of your second part, independent component analysis for patient classification?
Well, we are finalizing the manuscript right now, and it's going to be submitted, let's say, next month, hopefully. By the way, if there are some people who are interested in the method, they are welcome to contact us, and yeah, we can communicate by email about the methods. Thank you.
And Dr. Nazarov, it looks like we have time for one more question. How is your experience with the TAC software? Is it easy to use, and is it sufficient for the analysis?
Mm-hmm. Yes, in fact, I have some experience. The TAC software appeared last year in this current form, and in fact, we need to stop one of our draft preparation because of it, because we wanted to publish something related to the analysis, to the pipelines, but unfortunately, or fortunately for all the rest, this part was covered by the new tool. It is quite enough for the analysis, for the standard analysis of Affymetrix or Thermo Fisher arrays, let's say it like this. If you need something more advanced, then maybe you will need to use R afterwards, but it's very good when you need to load the data, to normalize the data, to see what's up there with experiments, are they reproducible, what are the outliers, and so on. And the nice thing, which I specifically like, is that the software allows for RMA with GC content correction, which is not so often, let's say.
Thank you again, Dr. Nazarov. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today will be addressed by the speaker via the contact information you provided at the time of the registration. We'd also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. This webcast can be viewed on demand through September of 2018. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.